So hello again, everybody who is still with us. We are at the final item on our program, a very exciting plenary talk by Professor Tracy Bale. Tracy Bale is a professor of pharmacology and director of the Center for Epigenetic Research in Child Health and Brain Development in the School of Medicine at the University of Maryland in Baltimore, USA, as well as the president of the International Brain Research Organization, IBRO, that we have mentioned several times already because they have provided us with support for several items on our program, and we are really grateful to have had their support. Dr. Bale's research focuses on understanding the role of stress dysregulation in neurodevelopmental and neuropsychiatric diseases, and the sex differences that underlie disease vulnerability in humans. So her talk will address the role of extracellular vesicles in stress signaling. I must add just one sentence, Tracy, before I letting the spotlight shine on you. In keeping with the other plenary talks that we have allowed some discussion after, if you are willing to stay with us a bit longer and answer any questions that may come, I would kindly ask you to do so.
Absolutely happy to do it.
Thank you.
Thank you so much, Maya, for the invitation. And I am incredibly delighted to be here, although I would be much more delighted if we could physically be together for this conference. And I had so looked forward to visiting your incredibly beautiful country. Unfortunately, we are not. We are on Zoom. But I think that also does allow us and individuals to join who may not be otherwise able to. So is it okay if I share my screen now, Maya? Yes?
Please do. Yes.
Okay. I don't want to cause any problems here. So thank you again for the invitation, and I'm happy to stay on, and hopefully there's still people who are interested in discussing science. It's the part I think I miss the most of this during this pandemic, is that our inability to be in physical spaces together and discuss science. Today I wanted to present to the neuroscience community some of the research in my lab that touches upon, I think, a lot of what you've been listening to thus far the last 2 days of this meeting. I know that there's a lot of interest in neurodegenerative diseases, but I think there's a great evidence that neurodevelopmental is also predictive of some changes in neurodegenerative. And so a lot of the same signals and biomarkers might be relevant. And as the talk you just listened to was discussing ways in which to identify precursors or biomarkers, I think that one of the aspects that goes across a lot of the projects in my lab is trying to understand those markers and move between our animal models and the human disease, and then translate those findings back to identify mechanisms back in the mouse. And thank you again, Maya, for the invitation. And yes, I am both with the research I'm talking about today, but I'm also president of IBRO. And so I'm here in both capacities. And I think it's exceptionally during the pandemic has made it clear how important global neuroscience and support of each other is. And so I am working very hard with the other members of IBRO, with the regional committees, as well as the officers, to ensure that IBRO resources make it out into the community to where the needs are around the world. So with that note, I'm going to talk about the two projects in the lab, and I'm going to focus on a concept that we've been working on for the past 20 years or so, which is trying to understand how the environment, in my lab, we model that looking at stress effects over different periods of time, depending on when they happen, who they happen to, and how they ultimately can affect how the brain develops. So many of you who work on animal models will appreciate that we oftentimes spend our studies looking at relatively healthy animals, whether they're rats or mice, living in vivaria, that they are well cared for, they are very healthy, they have access to very healthy food, they live in very healthy, clean cages. And so I think it is becoming more apparent that we need to spend some time thinking about what are we modeling and how does that replicate, who's at risk and why people are at risk. We call this the Jibaib Id. So what is your genetic background, where you come from, where your parents come from, those SNPs that place you at risk or resilience to perturbations in the environment, whether that you're at risk for neuropsychiatric or mental health disorders, whether you're at risk for cardiovascular disease, et cetera. And then the E is the environment. Are you raised in an urban environment where there's a lot of toxicants in the environment or a lot of trauma or stress in the environment? Are you raised in a rural environment where you may have a lot of exposure to other perturbations, such as maybe not access to healthcare? And then the D is developmental windows. And I think that's also very important to think about the timing of when insults may happen. And that when the insult happens may be a predictor of risk, for instance, in Alzheimer's or the neurodegenerative talks you've heard from in the last 2 days, that they may not present for decades, but the developmental window in which the exposure happened might be pertinent. And I want to add, importantly, across all of these factors of G by E by D, sex as a biological variable interacts at every stage, because there are many genes on the X and Y chromosome that could interact with other SNPs that can make you a genetic risk or resilience. Of course, gonadal hormones alter all three of these factors, but as well as how males and females may perceive perturbations in the environment or enrichment, for that matter, differently, as well as, of course, males and females, all mammals mature at different rates in utero and postnatally, over adolescence, and in aging. And so it becomes conceptually important to think about these factors when we model. So as I said, my lab is very interested in things that influence neurodevelopment. And you can't really think about a developing brain without first thinking about how mom and dad and their influences come together at the point of conception. So this slide is just a reminder to everybody who spends time thinking about aging or even just thinking about the brain, that the brain didn't exist until sperm and egg came together. And so it's important to think about the markers that come together. It's not just mom and dad's DNA, that there are many influencing factors that can alter the course and rate of development that ultimately can influence how the brain develops to be resilient or at risk. I also am reminded to point out from this slide, and we'll come back to this at the end when I talk about our paternal studies, that while one sperm here may be fertilizing the egg, millions of sperm carry signals to the egg and can interact with at the point of conception. So altogether, before I begin telling you about these two projects, it's important to think about that the developing brain is influenced by many factors, all of which, of course, in humans are very complex. There's the factors of mom's in uterine environment and all of the factors that she has experienced over her lifetime and may directly experience during different course of pregnancy. So that, again, those developmental windows, something that happens very earlier in the first trimester may have a very different impact on the outcomes compared to if it happens in the third trimester. And of course, as I said, egg and sperm coming from mom and dad, they bring preconception signals with them at the point of conception that are very important. And things that might just influence the biological intrauterine environment itself and how the biology of implantation occurs. We do lots of studies in the lab where we think about things like transplacental signals that offer cues to the developing brain about the environment it's being born into. So all of these factors, and again, depending if it's a male baby or a female baby, can be very distinct in those rates of development. Okay, so what are extracellular vesicles? My lab is very interested in identifying, once we understand models, for instance, of when perturbations such as stress can influence brain development, and trying to understand what the signals might be. Mechanisms are very important, but translating those into humans and being able to ask the questions about relevance to humans requires biological markers and being able to have access to measure them. So one of those that we've been looking at for the last several years is extracellular vesicles. Extracellular vesicles, you may refer to as exosomes. Exosomes are actually a small sized extracellular vesicle. So extracellular vesicle is the classic terminology for the general feature of these nanoparticles. They are secreted from all tissues in the body in all mammals. So your brain makes them, all tissues make them. The cancer biology field has really identified the most biological factors of extracellular vesicles and delivering signals in large part to the immune system. They communicate locally within tissues and they communicate systemically as well. So the brain, yes, can secrete extracellular vesicles into circulation, and similarly circulating extracellular vesicles can signal to the brain. We're interested in identifying if biologically extracellular vesicle components shown here on the left, where in this lipid membrane structure of these tiny nanoparticles contain tons of proteins. The composition of those proteins is very important for many reasons, which I'll get into. But the composition can tell you a lot about the biological tissues where these vesicles are being secreted from when they're traveling in circulation, and in part where they're traveling to and where they may act. The content, that the intercompartmental content of these nanoparticles contains a plethora of small non-coding RNA that are thought to act at these distant sites. So they are taken up by cells, they travel in circulation, they're taken up by cells in those other tissues, and these small non-coding RNA and proteins can have hosts of effects. They can immediately affect, for instance, translation of RNAs into proteins. It's one of the direct effects. On the right is showing you over the course of time, we've identified that these are not garbage. They are actually real signaling molecules back in the 60s through the 80s. And as we've learned more and more about these vesicles, the differentiation of them by size and the differentiation of them by the compartmentalization of where they are packaged within a cell and how they are secreted and where they act at distant sites. So they're really interesting particles, and you can learn a great deal of information by isolating and studying these vesicles. So I'm going to tell you about one study which was just very recently published in Biological Psychiatry. And I'm going to use this as an example as to how we think about these as biomarkers and why we think they may be functioning as a systemic stress signal. What's more impressive is that we are able to identify in this study an actual developmental insult that then is predicted by changes in these biomarkers many decades later in adults. So when asking about stress or trauma, this is in a collaboration with Tanya Jovanovic and the Grady Trauma Project at Emory. Tanya is now at Wayne State building another trauma project there. So in Detroit. And so we wanted to ask, if we ask about a very specific type of trauma, one that we know is more specific in terms of the timing in which it happened, in this case, sexual trauma to women, and we pulled data, so the Grady Trauma Project is able to recruit all individuals who come into the emergency room, and they capture biological information about these individuals. And so we asked, in an N of about 25 to 30 in each of these groups of these women are now in their late 30s and early 40s, if you had experienced a specific interpersonal violence of sexual trauma, if you experienced it early in your childhood, so under the age of 14, specifically during adolescence between 14 and 17, or as an adult, or no sexual trauma at all, keeping in mind that this is the inner city of Atlanta and that it is predominantly a Black population and is predominantly a population that has experienced a lot of overall trauma. Okay. And so we asked, looking at a host of physiological measures, could we identify specific risks of when the trauma happened, predictive of later life health risk, as well as we looked at a host of other factors I don't have time to get into today, including circulating cytokines and cell-free mitochondrial DNA. But we looked specifically at those extracellular vesicles by looking at both the small non-coding RNA content, but really focused in by proteomics on the protein content. Could we identify anything unique in these adult women decades after the trauma happened that might point to a biomarker of who is at greatest risk to know when and how to intervene and what they were at risk of? Okay, so we first looked at overall health of these individuals. I'm just showing you a little bit of this information, this data. So we first looked at predictive outcomes related to hypertension and diabetes. So as you can see, this data has all been analyzed by both grouping all of the sexual trauma women together, that's in the blue, and then breaking that out by the age at which the sexual trauma or interpersonal violence happened. So if you first look at, and this is not surprising, this has been shown before in interpersonal violence, that these women are at greater risk for what might be considered overweight, obese, or diabetes. They are increased body weight, increased hip circumference. They also had increased waist circumference. And if you break that down further by the timing, it seems to be largely driven by the women who experienced this interpersonal violence in their childhood prior to the age or prior to onset of puberty or adolescence, depending on, again, we can further break these data down to show that. So while sexual trauma overall seemed to put them at greater health risk, this data seems to be largely driven by a unique time in childhood when that violence occurred. Similarly, we see the same effect if we look at hypertension risk. So again, looking at green, where the high blood pressure category is increasing in risk for sexual trauma. Again, if you break it out, you can see, again, interestingly, earlier in childhood, hypertension risk is escalating. These are all, again, women in their late 30s, early 40s. And it seems to be progressive compared to during adolescence, compared to during adult only, compared to no sexual trauma. Overall, the data seem to point to, and this won't surprise you based on talks at this conference, that the trauma that occurred in earlier childhood seems to be aging these women faster. And that data has also been shown by Sierra Carter and others, that racial discrimination, stress, and interpersonal violence seem to progressively age, especially women, especially Black women. That data is pretty convincing. We also want to look for neuropsychiatric disease risk. So if we then looked at a host of things, I'm just showing you here the fear potentiated startle or fear conditioning, where we see something very different from neuropsychiatric disease. That again, there's an elevation overall for sexual trauma that seems to be largely driven by experiencing this interpersonal violence uniquely during the course of adolescence, that also fit with, and I'll show you in a second why this is important, looking at skin conductance in these women. So what I'm showing you in snippets of this data here is that when this trauma, this specific type of trauma occurred, seems to be predicted for different types of disease outcomes later in life. That the diabetes and hypertension risk seems to be unique to experiencing this trauma under the age of 14, whereas neuropsychiatric and mental health outcomes seem to be, especially related to PTSD, are more relevant to experiencing this during adolescence. Okay, what I really want to talk about is the biomarker. So as I said, we isolated from plasma samples from these women. We isolated extracellular vesicles. We then performed proteomics assessment on these, and I'm showing you the overall outcome here at the top. So this is comparing all proteins detected, showing you on the gradient in the control, no sexual trauma group on the top, showing you the high expressing proteins or high abundant proteins in these extracellular vesicles, all the way to the right, which is the very low or very low detectable. In comparison to the overall sexual trauma, you can see that there are clusters very much of proteins that are, whether highly abundant in the no sexual trauma, are very lowly abundant with sexual trauma group, and vice versa on the other end. Okay, so the interesting part is, what are these proteins and what can they tell us about the tissues that may have been programmed during that traumatic experience that might be interesting related to those disease outcomes. So we've now broken this down by, again, under 14, adolescents, or as adult in comparison. These are showing you differences related to the non-sexual trauma group. Some interesting things stand out. So this abundance of proteins here are showing you that are in the pink are the lowly detectable compared to no sexual trauma. What are those proteins? Interestingly, the abundance of those proteins, I'm showing you several of them here on the right, on the top, looking at log 2-fold change. These are really big differences in abundance of these proteins in this extracellular vesicles. And what you can see is annexins and galectins. And so for those who don't study vesicles, I'll tell you what those are. Those are really important key proteins in many tissues in the body that regulate the secretion of specific types of extracellular vesicles. And we'll come back to that. And these are very much downregulated. So what that's saying is, in specific tissues, you may be seeing a shift in the type of extracellular vesicle that is being secreted, and maybe signaling to things like the immune system or other tissues, putting individuals at risk for different health diseases. Okay, so looking at this blue are these very upregulated proteins in the adolescent group was the data I'm going to talk the most about today. So this is a unique subset, a large subset of proteins really only detected in this one group, the women experiencing sexual trauma between 14 and 17, not detected in the other two sexual trauma groups, and not detected in the no sexual trauma. So what are those proteins? Well, what was interesting is that those proteins, almost all, were keratin and keratin associated related proteins to the skin, which was very surprising to us. So in delving deeper into what they are and showing you in this bubble plot here, again, by the three groups, this middle group here showing you an abundance of proteins in extracellular vesicles in circulation, only detected and at high levels in this one unique group. Further interesting is that the majority of those keratin-related proteins, when you look at where they belong to, were almost all related to a 17q21 gene cluster on chromosome 17 in humans. Also of interest, and pointed out to us by our reviewers actually of this manuscript, was that similarly in this gene cluster are also CRF receptor 1 and microtubule associated protein tau. So both proteins very much associated with PTSD and the GWAS related to PTSD risk and very much important for stress response. So here we have a cluster of genes found in extracellular vesicles being secreted into circulation, pointing to the idea that something unique about the skin has been programmed during a traumatic event only occurring during adolescence. And as you might remember, is that the skin is your greatest barrier to the environment, and not surprisingly is highly innervated, and the maturation of this autonomic innervation occurs during adolescence prior to adulthood. And also of interest is that the HPA, the stress axis that exists in the brain, also exists locally within the skin. So there's a lot of overlap for how we respond to our environment, especially related to stress and activation of the autonomic nervous system that not only occurs in the brain, but also occurs locally within the skin. So altogether, what this data in an unbiased approach tells us that a unique and traumatic event occurring during a specific window of maturation point not only for the brain, but maturation for the skin, program differences in the EVs that are being secreted and maybe targeting uniquely and exacerbating the stress response or stress reactivity in these women. So a little bit more information about EVs. So extracellular vesicles, as I told you, are nanoparticles. They contain hosts of proteins that I just told you about. The small non-coding RNAs we're still evaluating from these extracellular vesicles. They also contain unique lipids that are becoming more of interest as to how they may provide unique signals at target sites. And so understanding about the vesicle size tells you a lot about the intracellular compartments of tissues and how these things are being secreted, and the differences between larger microvesicles and those very tiny exosomes. They provide different signals and cues to the and responses to the environment. And so understanding a shift in vesicle size may be very important for telling you what is going on following that programming event. So as you can see on the bottom here, we looked at the size difference of the median size using nanosite technology. Nanosite technology allows you to look by Brownian motion at when you load your extracellular vesicles from your groups to look at things like the vesicle size. They can tell you about the endocytic rate, the rate at which vesicles are being taken up, and the EV type, as I just said. So as you can see, we specifically see a significant reduction in those same adolescent exposed trauma women now in adulthood in their vesicle size, suggesting you're seeing a shift from a microvesicle containing composite to a more exosome containing composite. And that may tell you something about what's happening programmatically at the level of the skin based on our proteomic data. On the right, we can also use our nanocyte Zeta view machine to tell us about the zeta potential of these vesicles. So that tells you something about the surface protein charge and how it's interacting with the environment. And this is important because the charge tells you something about both the composition of proteins, the surface area of the vesicle, but also its stability in circulation. So the greater or the more negative the zeta potential of extracellular vesicles, the more stable they are because they will not aggregate and then be degraded, as well as the adsorption, the rate at which they're going to and how they're going to interact at their target. And as you can see, we also see a significant difference in this adolescent exposed group as to the zeta potential, suggesting a smaller size and a greater likelihood of aggregation based on the reduction in their zeta potential. So altogether, taking the data that I've shown you, there is something uniquely programmatic, likely involving the skin, and we're working now on identifying the unique, based on these proteins, the unique skin type, which seems to be a mechanoreceptor unique cell called a Merkel cell that seems to be interacting and maybe providing an integration point for PTSD risk that we're currently studying now in our animal models. But going back to the human, why do we care about this? Well, we care very much about women and experiencing trauma or adverse childhood events, such as interpersonal violence. We know that, especially cities like Baltimore, where I live, we have a majority Black city. The experience of discrimination, racism, and stress has a huge impact of how women will experience their environment across their lifespan and can be very important for predicting negative outcomes during pregnancy. Again, very much relevant to the developing brain. We know that African-American women in the United States are at much greater risk for for morbidity and mortality during pregnancy in the first year postpartum. Maternal death rates have not changed that much. They are 3 to 4 times more likely, the women and their infants, to experience mortality in pregnancy. So understanding what those risk population is very important. Extracellular vesicles are secreted in high concentration. In fact, the time in the lifespan where you can detect the highest number of extracellular vesicles is in the course of pregnancy, largely thought to be secreted from the placenta. So the placenta secretes a ton of extracellular vesicles into mom's circulation, no doubt interacting with the immune system, which is very important to interaction during pregnancy, but other factors and hosts. Yoel Sadovsky has done a tremendous amount of work looking at these extracellular vesicles and their signals interacting at the placenta itself, and can be very important for determining health outcomes and transplacental signals that tell the brain how to develop. So that's something we're really interested in, defining these as markers. Why? Because as I just said, the preconception environment or adverse childhood events are very important for understanding both for mom and the baby. So as a preconception event for outcomes that affect brain development. So the Kaiser Permanente study that determined these adverse childhood events that an individual can experience across their lifetime up until, well, especially up until adulthood, and that the unique feature of 4 or more of these adverse events prior to adulthood can be the greatest risk for predicting lifetime negative outcomes, including neurodegenerative risk, as well as all kinds of mental health consequences and addiction. So understanding the adversity of mom's experience prior to her adulthood and how it intersects at pregnancy is very important for understanding brain development. In the city of Baltimore, in comparison, if you look across the United States of America, currently pregnant women, the number of adverse childhood events that they experienced prior to adulthood and its intersection of pregnancy, that 4 or more point here in yellow is about 12.5% on average across the US. So that's still pretty high. But if you look for a majority Black city like the city of Baltimore, those women who have experienced 4 or more ACEs rises to over 60%. So what that tells you is we need to understand the population we're studying, the intersection of that adversity. So things like being able to measure changes in biomarkers is going to be very important for identifying who might be at risk and where we could intervene. So from our study, in summary, looking at things like unique identification of a specific skin type that could be sending signals of adversity or resilience. We don't know which way that might go. We're doing current animal studies to identify that. Could be really important for identifying women who might be at risk and following them more closely in pregnancy. And then understanding the intersection of things like promotion of neuroinflammatory or inflammatory cytokines, cell-free mitochondrial DNA, those signals that could alter the course of brain development. Okay, those are very important. So now I want to switch to an animal study that we've gone actually back and forth between the mouse and the human. But we've been able over the last decade to become very mechanistic in our questions and using the extracellular vesicle as a mechanistic outcome. So this is looking at our model of paternal, so the male's stress experience in his lifetime, and how the signals all the way down back to that sperm that I told you about earlier. What are the information that sperm bring to, what are the signals that sperm bring to the egg that can alter the course, just as simply as altering the rate of which conception happens, the rate of implantation, the rate of transition from 2-cell to 4-cell, for example, or to blastocyst stage, can be very important for the signals, because the timing of development, if you think about the sophistication of the brain, altering, for instance, in a mouse, your rate of development by even a half a day, or in a human by the course of weeks, could be very important. The survivability of a species may be enhanced, but at the risk to something as sophisticated as the brain, altering where the pruning happens, altering the different trajectories of the brain. And you may not see that outcome until decades later, for instance, in the case of aging and risk for things like neurodegenerative disease. The epidemiological evidence here is very strong. Does dad's lifetime stress experience, is that important? We know from many studies of looking at things like the Dutch hunger winter and the Swedish famine studies have looked over the last many decades and shown that yes, the male's experience over the course of his lifetime can be very predictive for multiple generations in terms of their longevity, aging factors, disease risk. So we wanted to understand mechanism. We developed a decade ago, more than a decade ago, a mouse model. I'm just going to quickly cover this. All of this work has been published. We use 4 weeks. There's a sufficiency of the amount of chronic stress that the male mouse has to experience. We then have done many different time course of breeding. It takes following the stress end, it takes an allostatic set point, meaning a post-stress resolution. It takes about 1 to 3 months for that resolution to happen, for the phenotype then to be passed on. If you breed these mice right after or during the stress, you don't pass on a phenotype. So there is a resolution that is required. We'll get back to that. The male and female offspring, the most important thing here is just showing you an example of how this stress is programmed at a reproductive level for this male mouse. He is able to pass on to his male and female offspring a change in how they react to, in terms of stress reactivity to their environment. And so I'm going to show you some mechanistic studies as to why we think that's happening. We've identified changes at the level of the sperm and level of the offspring brain. Okay, that's all published. So what does this mean? This is showing you an HPA, hypothalamic pituitary adrenal stress response in male and female offspring. This is showing you that if we do chronic stress in the dad or chronic stress double, meaning we've really hit this dad with stress, it doesn't matter. He passes on the same phenotype to his male and female offspring. Basically, they are hypo stress reactive as adults themselves. So they are reacting very differently to their environment after their dad was exposed to stress. And this is showing you 3 months after stress has ended, we bred that male, and this is what his offspring look like. And now it doesn't matter. We can breed that dad with many different females. We can breed that dad a month after, 3 months after. This is the exact same phenotype. However, if you look on the left here, if we breed that dad right at the end, so a week after stress has ended, he doesn't pass on a phenotype at all. This is showing you what happens with the single stress or the double stress, doesn't matter. The offspring look identical. So this requires an allostatic set point, but where? So we're looking at reproductive tissues to understand mechanistically where the signal lies. Okay, so this is just showing you isolating sperm a week after. So this is the model, 4 weeks of stress in the dad, breed him either a week after or 3 months after. Really no changes in dad's sperm a week after, lots of changes 3 months after. This has all been published. We want to understand mechanistically, what are we talking about here? Because if we can figure out mechanistically in the mouse, then we can ask more specific questions in the human. Okay, so for quick reproductive biology, this is a testicle in a mouse. We know that spermatogenesis occurs here, but the sperm are protected during the course of spermatogenesis by a blood testis barrier. So it's really difficult for factors in the environment to influence the course of spermatogenesis. However, once those sperm leave the testes at the end of spermatogenesis, they're not mature yet. They can't fertilize and they can't swim, but they're pushed through these ductals into the epididymis. This is called the head or caput of the epididymis. This is comprised of tubules in which the sperms spend many weeks maturing, experiencing bathing in growth factors to mature the sperm. And here they can receive cues, important cues from the environment. Those cues we think are epigenetic signals, but they're not occurring at the sperm. Rather, they're occurring in the tubules that align here. And this is just showing you an example of that. These are epididymal epithelial cells that line these tubules. These tubules are critical for secreting factors, including a ton of extracellular vesicles that interact with these sperm. And that is the mechanism that I'm going to show you about. We think it is these vesicles that are secreted from these epididymal epithelial cells outside of the testes that are experiencing and exposed to the environment, important signals that remain and can produce changes to vesicles many months after the stress has ended. Okay, so this brilliant graduate student, Jennifer Chan here, did a study where she developed a stress in the dish. Why? Because you cannot isolate extracellular vesicles in vivo in a mouse because these tubules are way too tiny. You cannot isolate the vesicles here. Obviously, these are not traveling in circulation. They're happening very locally. So instead, she spent the better part of years actually developing the stress in the dish model and perfecting it. So in this dish are DC2 mouse epididymal epithelial cells from the caput epididymis. And they secrete into the media extracellular vesicles that she then collects, and she can survey those extracellular vesicles and look for similarities in those vesicles to what we see in vivo. So she was able, over the course of several years, basically her thesis project as a pharmacology graduate student, to culture these cells, exposing them to stress hormones, corticosterone. She did a huge time course, dose response course, and this is shown here. So 3 years of her work is in this slide, showing you the rank analysis, rank hypergeometric correlation analysis, showing you a time course of cord exposure and concentration differences, and basically showing you that after 3 days of chronic administration and then washing off the court media changes 1, 2, 3 times and collecting the media to isolate the extracellular vesicles, she's showing you on the X compared to the Y-axis here that the similarities in those EVs in culture with the sperm in vivo, this is how this model developed. This is showing you the microRNA changes in culture, showing you that the stress levels are the most different and most similar to sperm. And really interestingly, the protein differences by proteomic assessment showing you in this heat map. So this is showing you vehicle treatment versus court treatment at this 11-day time point. This is the most similar to what we see in sperm in vivo. This orthogonal comparison here showing you immediately after, we don't see many differences protein-wise. By 11 days, we do see great differences. Again, most similar to the sperm. And as I showed you in the human study, we see a reduction in EV size using our nanocyte technology, showing you here that with court treatment, 11 days out, we see the greatest reduction in going towards an exosome type of vesicle. So this is all from in vitro. And now we're moving on in these studies and asking questions, well, what is it that is programming these changes? Because that's the mechanistic question. So at the level of that epididymal epithelial cell, how does it know it's stress? How does it know it's chronic in nature, and what is it changing? So on the left is in vivo assessment by Jennifer Chan. On the right is in vitro assessment by Nicole Nanyuk, that is showing you using cut and run, which is basically using an assessment of asking at the level of the DNA. It's basically a ChIP-seq assay. So I'm not going to go into all this because we don't have time, but what I want to show you is if we alter the glucocorticoid receptor on the left in vivo, specifically in the caput of the epididymis, or we ask where the intersection of the glucocorticoid receptor is with histone modifications, specifically a repressive mark, H3K27 trimethylation, in vivo and in vitro, we see strong evidence for thousands of genes and thousands of sites for the glucocorticoid receptor, chronically administer corticosterone, both in vivo and in vitro. This is telling you that there is something in the epididymal epithelial cell that is prolonged change. So that's that allostatic set point. It is dramatically shifted, and it is shifted permanently. We cannot reverse it. And that fits with this long 3 months out change that we see in what's happening with the offspring. Okay, so in vivo and in vitro, and all the way down at the level of the cell mitochondria, Nicole sees now that this change seems to be happening due to chronic glucocorticoid exposure using seahorse at the oxygen consumption rate, ATP production rate, and what the mitochondria even look like. So we know all the way down at the level of the epididymal epithelial cell in these animals, in vitro and in vivo, that what seems to be happening is a new allostatic set point is shifting what the cell is doing, and therefore what the cell is secreting. And I'm going to leave you here with this most exciting data. This is all recently published from Jennifer Chan's thesis. What is our evidence that anything that I just told you, even superficially, is meaningful to mechanism that we can explore in humans? Okay, so going back to her stress in the dish, she repeated those experiments. She collected the extracellular vesicles at that 11-day time point, either from vehicle treatment or court treatment. So she has 2 pools of vesicles. She then took a male healthy mouse. She took sperm from the caput of the epididymis. She divided it into 2 pools. She incubated the sperm from this one dad, either with those vehicle treated vesicles or prior court treated vesicles. She then washed the sperm, we performed ICSI, or intracytoplasmic sperm injection. Now, this is eggs superovulated from one mom. So sperm from one normal dad, eggs from one normal mom. The only difference is what those sperm were incubated with, these EVs. Performed ICSI. We then were able to then take a donor mouse. So we then transfer these zygotes into either the left or right side, so the same environment. Okay, so sperm same dad, egg same mom, transfer into the surrogate same environment postnatally. In utero. So we did two different types of experiment. We either then looked at mid-gestation or embryonic day 12.5, and we asked, using transcriptomic approach, are there any differences? Does the signal, largely the protein signals from these EVs, do they matter? Do they matter to the course of development? Do we have any evidence, perhaps, that the rate of brain development is being changed? We also performed another set of experiments where we let them grow up and asked, do we see evidence that their HPA stress response, similar to the paternal stress dads, has been changed? And the answer is yes. So in this very complicated experiment at the end of her thesis work, transcriptomically, as you can see here with gene set enrichment and the Go terms analysis, we see dramatic differences actually in synaptic signaling and neurotransmitter transport. What that probably is telling us is that, in fact, the rates of development, and this is the evidence that we see from this model, that some signals, some protein signals in those EVs from court treatment or from stress is telling the development rate to speed up. And in fact, we see that the rate of implantation is enhanced in the zygotes that were from the court treatment. So this is not surprising, as you know, from stress in the environment for things like famine or war. When the stress is resolved, species needs to rebound, and those signals evolutionarily probably are preserved to speed up the rates and survivability of a species at the risk potentially of the developing brain. And the bottom experiment here in E, what I'm showing you is that, in fact, what we saw was that corticosterone in an HbA assessment of these adult animals, similarly to in vivo work from the paternal stress, is in fact reduced. So all told, those signals seem important. And we're now working using fibrophotometry to ask what is different in those brains. I don't have time to go into those studies. So I just want to end in the last 2 minutes by saying, how do we translate this into humans? Humans, of course, are much more complicated. We are not rearing them in healthy cages with high fiber, high enriched diets and clean environments protected from predation. We're humans and we're very complicated. So when we started thinking about how to assess this, it occurred to us that no one had asked the question about even the sperm content and the small non-coding RNAs, which I haven't spent a lot of time talking about, but wanting to ask, what do we know about human sperm and the diversity of the composition within subject over time and across subjects? Okay, so in a collaboration with psychiatrist Neil Epperson, who, when we were both at the University of Pennsylvania, we did this study. She is now chair of psychiatry at University of Colorado. So we wanted to ask, if we assessed young men on the University of Pennsylvania campus, a relatively homogeneous population, if we assessed based on perceived stress in the month prior and had them come in and donate 6 months in a row, doing a full battery every month of perceived stress and adversity experience, if we assessed that and then modeled that by collecting their sperm and sequencing all of the small non-coding RNA present in sperm, including microRNA, tRNA fragments, and piRNAs. And then we did a bioinformatic modeling of all of this data. Could we find anything consistent with which low expressing RNAs consistently or high expressing RNAs consistently are the most interesting? Those that were dynamic, meaning from month to month, seemed to be variable enough to model with the perceived stress. Okay, it was a big question and a lot of complex data. So I'm showing you a little snippet of it here. This is also all published. So by column is the subject. I'm just showing you a snippet of this data. This is just 5 men here in this study. We had 21 men. So subject numbers on top, 13, 2, etc. This is subject 13 sperm over the course of 6 months. MicroRNA is the rows. So microRNA at top, tRNA fragments in the middle, and piRNA in the bottom. So with the modeling, I'm showing you the dark blue are those that statistically rose out as being dynamic for each population. And if you look closely, the light blue are actually all of the background microRNA, tRNA, and piRNA. So that's not a blue background. Those are actually all of those small non-coding RNA detected. Okay, so what I'm showing you here is that those that rose out between subject and within subject is statistically different in relation to stress. I'll show you in a minute. So I think I want to point out just something really interesting, which is that the piRNA and the bottom row here also seem to be cycling across men, which has never been shown before. So very interesting. And I'm just going to end here on those that fit our model of prior stress, and it ended up being time plus 2. So if you had a high level of perceived stress in month 0, these dynamic microRNA and tRNA and no piRNA made that cutoff, were statistically different to be elevated by 2 months later. And most fascinating is that 3 of these microRNA that we identified within and between subject are already known to be important for reproductive outcomes in humans. So stay tuned for what those might mean in relation to brain development. Those are studies we're doing now. So I'm just going to end there because we're out of time and say that timing of specific trauma, as you saw in our studies, with its relation to reproductive outcomes or identification even of the interaction of skin level, that EVs, the type of EVs and where they may be signaling from and to is very important. And the EV content is something we're very interested in. And I hope that I've convinced you that extracellular vesicles in all types of disease states, there's studies now that are coming out looking at systemic EVs in relation to neurodegenerative diseases as well, and aging, very important biomarkers that might be related to adversity earlier in life. And lastly, but most importantly, to highlight the individuals that have done all of this work. As I said, Jennifer Chan was a graduate student in the lab. She's now a postdoc with Ian Mays at Mount Sinai. Nicole Kanyuk is the MD PhD graduate student working on following up on these studies. Chris Morgan is a postdoc in the lab who's done all of those human assessments and all of the very complicated bioinformatics. And Katie Morrison, in working with the Grady Trauma Project, was the postdoc in the lab doing the human studies related to looking at the EVs in circulation with interpersonal violence. Of course, our amazing collaborators with the Grady Trauma Project and Tanya Jovanovich's group, and all of our collaborations when we were at Penn and now at Colorado with Neil's group, and understanding the importance of EVs and reproductive outcomes. And of course, all of our NIH funding to support this research. Please follow us on Twitter. We do a lot of interaction with the community, and especially with, as being president of EBRO, around the globe. And with that, Maya, I'm happy to take questions.
Fantastic. Thank you very much, Tracy. It was really rapid paced, but also very rich talk that gives a lot to think about. And we do have a number of questions. Let me start from the most basic. Did you measure the contents of corticosteroids in EVs from in vitro experiment? Is glucocorticoid carried by EVs?
We did not, because steroid hormones, because they are so lipidophilic, are not easily stored anywhere. They so rapidly travel in circulation that the steroid hormones themselves are unlikely to be a composition of the vesicles. But related to the steroid hormone processes, I think something interesting that has been a little more difficult to assess is the different types of lipids in those vesicles, and that's something we're working on. The lipid content is something that you have to, as an omics approach, you have to have a large number of them to assess. But I think related to the steroid hormones, it's a great question.
And then we go to out of the petri dish into the more the wider view. Did you also check for PTSD diagnosis within the traumatized sample to confirm whether this subset specifically shows more expression of the skin-related proteins?
Yes, so this is a great question. So what isn't clear, because of course I whipped through it so quickly, is that all of the plasma samples and the physiological readouts were actually a process of data mining of a cohort that was collected, right? So the Grady Trauma Project, everybody who comes into the ER, they try to recruit into and capture their information. So they do all kinds of surveys, they get plasma samples. Many of them will then participate in fear conditioning studies, et cetera. So we did not prospectively conduct the fear conditioning studies, rather we looked at the data. Future studies are now doing that exact recruitment. So that's a great point. So because we were doing it retrospectively, in relation to the EVs that we then assessed ourselves, was we controlled for PTSD symptoms across all of the groups. So unfortunately, we couldn't ask about PTSD risk in a unique group because they were equally distributed across all groups. But it's something we're very interested in now and asking prospectively if we identify a group with EV changes and then look at fear conditioning outcomes. Yes.
Then what I see are some, okay, given that much or most IPV, interpersonal violence, occurs within families or individuals' inner circles, can you really separate effects of interpersonal violence events in early life from the effects of long-term exposure to a threatening environment?
That's a great question. It's a question as to, that was why we selected interpersonal violence, because you can so more specifically identify the timing of when it happened. So we excluded women that had experienced, for instance, if you had been molested across windows as opposed to during a unique window, right? So that made selection of our individuals more complicated, but identified the unique windows. But yes, I mean, how could you possibly say that somebody who was molested as a child didn't then experience other types or continuation of types of trauma? It's a great question and a very complicated question for sure. So we only assessed that type of interpersonal violence uniquely for that study, but how it might interact with other types of trauma during different parts of development, absolutely, we agree that that's something that's a very complicated question. Despite that, I mean, clearly these unique proteins were only detected in that one group. But what that might mean for the risk or what's happening during that window and how it then affects interactions, let's say later, we don't know. All studies remaining to be done, but very good question.
This probably ties into that as well. How culturally dependent are these responses? For example, certain environments accept corporeal punishment. May the events of interpersonal violence by victims be perceived differently and have different effects than by victims in alternative environments?
Yeah, for sure. I mean, these are all really, really great questions and things. As we just submitted a huge grant to assess some of this that we thought about. In the United States, racial discrimination for African Americans is a huge question of over the lifetime, the effects that it has on stress reactivity and mental health. And so that was something that we were focused on for our NIH grant. But yes, globally, I could imagine all types of local, geographical, cultural aspects that would be really important to consider and incredible questions to be asked related to prediction of health outcomes as well as biomarkers, for sure. Yeah.
And finally, the question that touches on what we can translate from that research into the everyday life at the interface of clinicians with patients, right? So do these results mean that clinicians should ask patients about their parents' stress experience as part of the family history taking to gain a better insight about possible risk factors for any specific disease they may be suffering from?
I think that would be ideal, but the likelihood, I think we've come, I don't want to say a long way because we have a long way to go when it comes to mental health, certainly, and understanding it. But I think, when you sit down in your doctor's office, at least in the US, they will ask you a health history, right? They will ask about cardiovascular disease in your family, cancer in your family. They're getting better about asking about mental health outcomes. It is now required in the US when you sit down in your doctor's office that they give you sort of your own mental health current state. So, would a next step be to ask about more adversity for parents? I think it's logical. I think mechanistically, we need to understand more. Animal models and in vitro models, I think, are helpful for those mechanisms, and then figuring out how they translate so that we can ask better questions, certainly, to identify what does translate and how we might identify risk. Certainly during the pregnancy state, which is such a high metabolic demand state for women. We know that, for instance, in Black women being at the greatest risk, regardless of socioeconomic status, that identifying what those markers are could be incredibly helpful to physicians. So could that next step be to ask more, not just mental health questions of our parents, but ask about what other adverse experiences for all health outcomes could be important. I think we have a long way to go for mechanism, but we need to get there.
Yes, I was just thinking about how that would really change the way that we conduct interviews and take histories, because it would be a shift from rather matter-of-factly assessing, specific illnesses, making an inventory.
Yeah, in Slovenia, when you sit down with patients, especially new patients, and do you for a health assessment, do you ask about parental or familial mental health?
So it would be unfair for me to claim to know as much as I should probably about that, because even though I am a physician, I'm not a practicing one, so I'm not in clinic. But I suspect it is part of every history taking to assess any illness that has been present in the family, including mental health issues. But I don't think that anybody, speaking strictly about physicians, right? Any specialty. So that anybody really goes in that direction very far.
What's interesting is in the Netherlands, because the Dutch hunger winter has prompted so many inter and transgenerational studies that they seem to assess that in a very different light, I think, than a lot of other, certainly more than the US would. The US is such a mixture of people coming from all over the world, but certainly when you interact with individuals from the Netherlands, it seems very obvious to them that adversity is important for predicting next generation's, health. That is something that they think about. So I think, as we learn more, I think it might become more part of our vernacular to think about.
Mm-hmm. Excellent. I think that's it for the questions. I don't think I've missed any. I do have to go back to your initial slide, because you were so sweet to include a Slovene greeting. Zdravo was perfect. But the other one is, instead of saying the way that we would spell it, dober večer, which means good evening, you said sober večer, which means, like sober, stay sober tonight.
We have to blame Google for that, Maya.
That's
well, if it's been sober. Stay sober.
Yeah, so far it's been sober. But we are really at the end of the program. So we are about to close it. And I am really grateful that we have been able to have this phenomenal presentation for the end. And I'm also grateful for the opportunity to pose questions to you, because before it seemed that there wouldn't be any, or that we could not address them. But thank you, really.
Thank you, Maya. And if anybody has any other questions or things that they want to, discuss, I'm really easy to find. You can email me at any time.
Thank you.
Thank you. Thank you, Maya.